can't see our faces, though.
Okay, well, yes you can.
Okay.
I remember the night before I started. I couldn't sleep, I was so nervous. I finally had a big girl job. I was about to enter the vast world of research, and I had no idea what to expect. What if I mess up? But what if I cure cancer? This summer, I had the opportunity to research at the University of North Dakota Medical School under Professor Scott Garrett. This project was funded by the NIH and has the potential to have an impact in North Dakota because of the exposure that our state has to cadmium. This summer, I studied the gene TRP10 and its expression in MT3 transfected and cadmium-treated human proximal tubule cells. The purpose of the study was to determine mechanisms behind epithelial to mesenchymal transition in proximal tubules during cadmium toxicity, mesenchymal to epithelial transition induction after MT3 overexpression. TRP10 is thought to be EMT promoting. So in this presentation, I will summarize my work and the conclusions that I came to this summer. You're probably wondering what in the world I'm talking about, so I'll start with a little background. Cadmium is found in the environment and has been known to cause a lot of health issues. In North Dakota specifically, there's a lot of cadmium in our environment because of all the farming. The kidney is the main site of accumulation of cadmium, and more specifically, it can be found in the human proximal tubules. Chronic exposure to cadmium has been known to cause kidney disease, which can lead to EMT. So what is EMT? EMT is epithelial to mesenchymal transition, and epithelial cells are like your skin cells, and they transition into mesenchymal cells by losing their cell polarity and their cell-to-cell adhesions. Mesenchymal cells acquire more migratory and invasive properties. So you can see in this figure, they start out as epithelial cells, and they're more square-like and they're adhered to each other. And during the transformation, they turn into mesenchymal cells, which acquire different shapes and migratory properties. EMT causes fibrosis, which can ultimately lead to chronic kidney disease. Fibrosis occurs when inflammation heals a scar tissue. So here's a figure describing fibrosis. And you can see here that when the cells are damaged, they heal more as scar tissue versus how they were before. This lab has done previous work in heavy metal exposure to the body in metals such as cadmium. My work involved 3 cell lines. My first one was HK2 cells. These are immortalized human proximal tubule cells, and it included another control that were HK2 cells transfected with 3.1 and HK2 cells transfected with MT3. My second cell line was acute cadmium exposure. These are HPT cells that were exposed to cadmium for 24 hours at various concentrations. My third cell line was chronic cadmium exposure. These were HPT cells that were exposed to cadmium for 13 days, at various concentrations. This lab has previously found that HK2 cells lose their expression of MT3 during the immortalization process. So in order to allow these cells to express MT3 again, the cells need to be transfected with the gene via a vector. And our lab specifically used this vector here. So this is a picture of the empty vector, and the gene MT3 was transfected into it, so we could see an expression of MT3 again. Cadmium is known to cause kidney damage. It can lead to fibrosis, which is mediated by EMT. My job was to test if cadmium can cause EMT when in HPT cells using a specific gene. My gene was TRP10. TRP10 is expressed in kidney cells, and when it is exposed to the TGF-beta factor, expression of TRPC10 increases in renal fibrosis. TRPC10 induces extracellular matrix decomposition, which leads to fibrosis. So the hypothesis that I came up with was that because TRPC10 induces EMT, when in MT3 cells, it should be decreased, and in cadmium-treated cells, TRPC10 expression should be increased. Unfortunately, my results were inconclusive. My results showed that TRPC10, when overexpressed in MT3 cells and compared to the HK2 cells, there was a significant change, but not when compared to HK2 3.1. And as you can see here in this graph, the HK2 MT3 is the only one that showed significant change. In my 2 cadmium cell lines, I had negative results, and I did not find any significant answers. So in order to make an accurate conclusion, more testing needs to be done. So my conclusion came to that because of my poor slopes and efficiencies when testing my gene, I need to do more testing. This testing would include more PCR, which is also known as polymerase chain reaction, to see how my gene affects EMT in the kidney. It is definitely frustrating spending an entire summer working on a project that I became so invested in. But it wasn't worthless. I learned more in those 10 weeks than I have in the last 22 years. And I might not have cured cancer this time around, but hey, welcome to science.